Hello, everyone. Welcome to today's webcast. It's 10 a.m. here in Montana, so we're going to go ahead and get started. I am Cheryl Rogers, the Director of Marketing here at Golden Helix, and Bryce Christensen, our Director of Services, will be presenting today's webcast surrounding population-based DNA variant analysis. Bryce, the floor is yours.
Okay, Cheryl, thank you for the introduction, and I'd like to welcome our audience here today for today's presentation. Today we're going to, as Cheryl mentioned, talk about population-based analysis of data coming from next-gen sequencing platforms. We'll discuss primarily rare variant analysis, but also talk a little bit about some of the things you need to know when it comes to analyzing the common variation that is identified through sequencing experiments as well. Now, as we go along here today, if you have any questions at all, feel free to type those into the question panel in the GoToWebinar software. We'll try to answer as many of those as we can at the end of the presentation today. Just a little bit about our company, if any of you are not familiar. We've been in business since 1998, providing both analytic software and services in the genomics industry. Last time I checked, we were closing in on about 1,000 citations in published papers, and you can find all of those listed on our website if you want to learn a little bit more about how people are using our products. Our flagship software tool is the SNP and Variation Suite, or SVS, and as we go along today, we'll spend quite a bit of time both interactively looking at SVS, but also looking at some results that were generated using the SVS software. In general, I would describe SVS as a platform for analysis, management, and visualization of many different types of genomic data. Today specifically, we'll focus in on DNA sequence analysis. So our agenda today will go something like this. We'll start out by talking just a little bit about what rare variant analysis is all about and what makes it tricky. We'll talk about some of the different options that are available for rare variant analysis. Then we'll move into SVS and talk about how to build workflows for rare variant analysis and, more generally speaking, for DNA sequence variant analysis. Then we'll look at some of the methods that are available and compare and contrast the outputs a little bit and talk about how they behave in different scenarios. Then we'll wrap things up by talking a little bit more about upstream analysis, things you need to know in preparing your data for this type of work. We'll also talk a little bit more there about common variant analysis methods. And hopefully we'll have some time left for questions when we're done. So what is the problem here that we're trying to address? A lot of us in the field, myself included, have quite a bit of background and experience working with array-based GWAS methods. This has been really the primary technology for gene finding research for the past decade or more. Over the last several years, gradually that's been swinging more towards sequencing-based technologies. Now, the NGS technology, especially whole exome sequencing, which is being done in very high volumes right now, makes it possible to include rare variants in an analysis. So with GWAS, the SNPs on the chips, by definition, tend to be common features. And with the sequencing, we can start to explore the space a little bit more and see what else is out there and try to incorporate it into our knowledge of whatever disease or conditions we're studying. Now, from a statistical perspective, those rare variants, of course, lack power for standard GWAS approaches. So if you have 1,000 samples in an experiment and you have a particular variant that's only in 2 or 3 of them, even if they are all 3 affected individuals with the disease, a typical contingency table-based test is not going to pick that up, even if it is a causal disease-causing mutation. So we have to be a little more creative in terms of how we utilize the information coming from rare variants. The standard approach that has been in practice for several years now and continues to be developed is combining rare variants into logical groups somehow and analyzing them as a single unit. Most often what we see is genes being used as the analysis unit. So we're looking within genes, assuming that any variant within a particular gene should have similar influence on the trait of interest. And so we can collapse that information or look at the burden of variation gene by gene to identify trait associations. Now, there's different approaches out there. We see some people preferring more of a direct search for susceptibility variants, relying heavily on bioinformatics. Typically with smaller sample sizes, you might be able to do this. Perhaps you have 5 or 10 samples or a single family where everybody has a very well-defined phenotype that's expected to be a Mendelian sort of disease. Then you might be able to quickly identify a gene that has variants in all of the samples and have a really good candidate without doing too much in the way of statistics. Although more often what we see is this approach being used with complex diseases with larger sample sizes that may be several hundred, several thousand, tens of thousands of samples, where we're looking for perhaps more subtle effects. So there may be many, many genes involved in the disease, and each contributing gene may only have some small subset of the samples that are carrying disease-causing variants within those genes. And so this requires some more advanced statistical analysis. There are 2 major families of collapsing tests depending on how you slice things up. Here it's based on a paper from Dan Shade from Genetic Epidemiology a couple of years ago, where they're defined primarily as either burden tests or kernel tests. Burden tests, simply put, combine all of the minor alleles across multiple variant sites, and there are different subcategories there. There are burden tests that operate without weighting. This includes the CMC test, which is in SDS, and we'll talk more about it in a few minutes. There are other burden tests that apply fixed weights that are based on allele frequency. There are tests that have more of a data adaptive weighting procedure like KBAC, which weights variants according to their prevalence in cases versus controls to a degree. And there are other methods that have data adaptive thresholds based on allele frequencies and so on. And then there are numerous extensions to all of these methods, sometimes perhaps to allow for effects in either direction or for other disease models or testing other hypotheses. The kernel tests, really the family of methods revolves around SCAT. The SCAT method has numerous extensions available for it. One of them is SCAT-O, which is a method that was just incorporated in the latest release of SDS, a method that we'll be talking about quite a bit today. So within SVS, the major methods you'll see in terms of the burden tests are the CMC method, which is unique because it is a simultaneous test for association of both common and rare variants in a gene. Most of these burden tests are really focused on rare variation, so CMC is kind of special in the way that it's able to incorporate more common variation. Within SVS, there are 2 options for running that. There's the standard method that uses more of an asymptotic distribution for determining significance. There's also a regression-based method that allows you to incorporate additional covariates in the test. Now, the KBAC method is also available in SVS. It's very much focused on rare variants. The test models multi-locus genotypes across gene regions and calculates an association test based on that. The standard method uses permutation testing, but we also have a regression method or a mixed model regression method available for KBAC. The mixed model method is especially unique. It's something that we gave an entire presentation on last year, which you can find recorded and available on our website. So SCAT, what is SCAT? What makes it different? SCAT, I suppose the simplest way to describe it is to say that it calculates a test statistic for each individual variant observed within a gene and then aggregates all of those variants together to compute a gene level or region level p-value. As I mentioned earlier, there's a lot of different extensions of the SCAT method to account for different study designs. A couple of quotes from a review article on SCAT. The method can be more powerful when causal variants have bidirectional effects and or a large proportion of the variants within the gene are non-causal. So that makes it a little different from some of the burden tests where there's an expectation that all variants in a gene are pushing the phenotype in the same direction. SCAT's able to tease it apart and say, no, there are variants here that favor controls, there are variants here that favor cases, and they're both important. But you see there, that last bullet, SCAT's believed to be less powerful than burden tests when the effects are unidirectional. Which brings us around to SCATO. SCATO is an optimized SCAT approach which is intended to combine the SCAT test together with a burden testing model in sort of a unified approach. So it adaptively determines how much to weight the burden test versus how much to weight the SCAT test to give the optimum result at a gene-by-gene level. So SCATO, as I mentioned, was just implemented in the most recent version of SVS. And we'll take a look at it here in just a minute. But one of the interesting things that you'll find as you look at the SCAT tests is that, like many of the burden tests, they do incorporate a weighting procedure, variant by variant. And the weights that you apply can have quite a difference on the results. What you see here on the screen, this red curve, represents the default weighting scheme used in SCAT according to the allele frequency of the variant, and you'll see that the rarest variants get much, much higher weight in aggregating the results than do the more common variants. You can compare that to something like the more uniform weighting approach, which is also an option available in the SVS implementation, or this black line, which is based on the Madsen and Browning weighting procedure used in the weighted sum of squares statistic that's one of the earlier burden tests. So you can see that there's really some major differences in how you might treat rare variants versus common variants in these tests. The question comes up fairly often, well, which one's the best? What should I use? And what I would say there is to keep in mind that the different weighting procedures are really testing different hypotheses. And so it all comes down to whether you want to identify genes where the rarest variants are expected to have the greatest effect, or if you want to take, say, a more uniform approach and just look for any combination of variants that seems to be associated with disease status. So we're going to move ahead now and go into the SVS software and see how this all works. We'll look at it a couple different ways. To begin with, we'll open up some data, look through it more in an exploratory fashion, and just see how SVS allows you to visualize this data, and we'll simulate the development of a burden test. From there, we'll look a little bit closer at how to do a formal rare variant association testing workflow for some of these different methods. Now, what you'll see in SVS is that it's very flexible in terms of workflow design. It has many different tools for data manipulation, for statistics, for summaries, for variant annotation. These things can all be used together to guide us through the data. As long as we have some idea of where we're starting and where we want to go, we can usually put together a workflow to get there. Now, the data that we'll be looking at here comes from the 1,000 Genomes Phase 3 release. With Phase 3 of the 1,000 Genomes project, we have whole genome data available for over 2,500 individuals. From that data, we went through a simple process to simulate a phenotype based on asthma. Asthma, of course, is a complex disease with complex genetic biology underlying it. What we did essentially was to create a simple definition of what a loss of function variant would look like. It would be, in this case, a rare variant predicted to be damaging by at least one of 5 predictive algorithms. There are 395,000 such variants scattered through the Phase 3 data. And then we just selected 8 specific genes that have been previously implicated in asthma, and if a sample carried one of these loss of function variants in one of those genes, it would, in most cases, not always, we allowed for some variable penetrance to be affected. So if you really look at what the simulation looks like, we definitely took a rare variant approach with this. So starting here with this first gene, IL-12B, there are a total of 18 rare non-synonymous variant sites based on our definition, 11 of which met our loss of function definition. 19 total samples have one of those loss of function variants. We made all 19 of them cases, but that represents less than 1.5% of all cases. So this is a situation where we have a highly penetrant disease gene, but it explains a very small proportion of the disease in our model. Moving down, we see some of these that have a much higher proportion of all of the samples, nothing that's more than 5.5% of the samples carrying a variant there. But we also see reduced penetrance in some cases where not quite all of them are diagnosed. So what we need to do then is figure out how we can find these genes where just a handful of samples are carrying potentially disease-causing variants.
Okay, let's go ahead and close out of the slides now and take a closer look at SVS. For those of you who might be seeing SVS for the first time today, I'll just point out that SVS is a project-oriented software package. It's designed to help you organize all of the inputs and outputs and intermediate steps of an analysis workflow in one place, together with any visualizations that you might create along the way. I'll begin by opening a project where I already have some data for us to look at. So inside the project now, we see what we call the Project Navigator view. There are several icons here representing individual data sets that have been brought into the project, or in this case, a visualization that was created previously. Let's start by opening this first one and talk about what we see. So here we have a tabular representation or spreadsheet representation of all of the variants on chromosome 16 in 1,000 Genomes Project Phase 3. This was created by reading in and processing the VCF file downloaded from the 1,000 Genomes Project. As we look at this spreadsheet, we'll notice that there are about 2,504 rows, so one for each sample in the data. If we scroll out to the end, we see there are about 2.7 million data columns. There's one column for every genomic coordinate where at least one sample carries an alternate allele, a genotype that is different than the human reference sequence. Also, you'll notice that there's an embedded marker map feature here that allows us to capture some annotations about each one of these variants, where we can see the chromosome it's on, the base pair position, the RSID if there happens to be one, reference and alternate alleles, and then it's possible to capture a number of other parameters here if you like to do so. Now, as I'm exploring data for the first time, I often like to see it visually. And one of the nice things in SVS is that I can just click a button here, and launch the Genome Browse visualization environment. And here within Genome Browse, if we go directly to chromosome 16, where the data is, what we see to begin with is essentially a density plot, giving us some idea about where the variation occurs within the data. So darker colors indicate a lot of variation, lighter colors, little or none. Of course, we have a large centromere region here on chromosome 16. Now, if we pick a region and just zoom in a little bit, we'll see that the image updates. And now we get this very colorful picture showing us the exact variants. And so it's color-coded, where A variants are always one color, C's are always another, and so on. So we can recognize exactly what is going on in here. Now, as we look at the data, we can recognize there are some variants that are very common, others that are very rare. And sometimes it might be helpful to look at it more from the perspective of a grouping variable. Now, because this is 1,000 genomes data, I happen to have the 1,000 genomes population information here. And by selecting the grouping variable now, it will update and reorder all of the rows so that now we can see there are certain variants that group together. So this one here at the bottom is very common in the red group, which is the African populations. We can see right up over here a variant that appears to be very common in the green section, which is the East Asian populations. Over here a little bit further, there's one that looks like it's common everywhere except East Asia. So it's kind of interesting to see those patterns. But of course, it's more often that maybe you'll have a case control variable, which will allow you then to look at controls and cases and see if there's any particular pattern for any of these variants. Now, what we're going to do is walk through the process of how you would approach a rare variant association test. We'll look at it both visually and analytically here. But to begin with, if you're looking at this, you'll recognize, obviously, there are some very common variants. If we're doing a rare variant association test, we need to perhaps remove some of those. Also, we may want to focus only on variants that we expect may have some influence on the gene product. And so we'll need to decide how to approach that. SVS allows us to interact with a number of different annotation sources for looking up parameters of individual variants and applying filters to keep or remove certain types of variants. And as an example here, if I want to visually look at a database of functional predictions, or perhaps variant frequencies, or even just look at the reference sequence, it's easy to add those into the image where I can see them. And let's adjust this just a little bit so we can see everything on the screen. And go over and take a look at the IL-27 gene. So as we look at IL-27, again, we see some common variants, we see some rare variants. If we pick something and take a closer look at it, for example, here in exon 2, we see what appears to be a very common variant. It's in yellow. Yellow tells you that the alternate allele here is a C. If we look a little bit closer, we can see that it is in the ESP database, where it's recorded to have a frequency of about 5% in European Americans, a little bit higher in African Americans. We can also look at it functionally and see that the DBNSFP resource tells us, it looks like this is non-synonymous. It's a serine to alanine amino acid change. But also it appears to be likely tolerated. It's not going to have much influence on the product of the gene. But as we look around, we may find some variants that are rare and or predicted damaging. So just over here in the next exon, let's take a look at this one. Here we see a variant that appears to be very rare. It's not captured in the ESP database. And if we look at the functional predictions, a G to A change at that location, which is what our variant is a G to A, we can see, again, it is non-synonymous. We see what the amino acid change is. This one, though, is predicted as potentially disease-causing or probably damaging by a couple of the methods. They don't all agree, and that's not unusual, but it gives us something to go on. So what I'd like to do is back out of this and open a different spreadsheet. This is one that was filtered in advance only to show the exon regions of chromosome 16. You'll notice that the data is much smaller now. Only 50,000 out of the 2.7 million variant sites on the chromosome occur within exons. But if I want to do a rare variant test on this type of data, I would start by identifying the profile of variants I want to keep. And what we'll do is just replicate the data simulation and get the exact same types of variants that we simulated to be loss of function. To do that, I can use the annotate and filter variants option, select the data sources that I want to filter against. In this case, I'll filter against the functional predictions as well as common variants from the dbSNP database, and then we'll also look at the ESP frequencies. So the software now is going to prompt me to determine how I want to use each one of those. And for the ESP data, we'll apply a filter based on the frequency, so we'll remove anything where the frequency in, we'll go ahead and use Europeans, the default, is greater than 1%. Then, based on this list of common SNPs derived from dbSNP, we'll remove anything that's in that common SNP list. And then finally, we'll keep the default filter on dbNSFP, which is going to remove anything that's synonymous. It will remove anything that is non-synonymous but uniformly predicted benign by all methods. So it will just leave us with things that show potential for being damaging in at least one method. So this will take just a moment. It's on the last stage of the filter. When it's done, we'll get a report of exactly what was found. And here's our report, so we know how many SNPs fell out at each step of the process. Then we get this filtered list where there are about 19,000 variants remaining. As we look at this list now, maybe we want to learn a little bit about how many variants there are in each gene at a sample by sample level. And to do that, we would come to the DNA-seq menu. Under this collapsing methods submenu, there's an option to simply count variants per gene. This option is really useful. It allows us to either look strictly at heterozygous variants, strictly at homozygous variants, or to consider both. We'll go ahead and consider both of them. And then create an output table where in a gene by sample table we can see just a binary indicator of whether or not the sample has a variant in the gene, or we can count the number of variant sites, or count the number of alternate alleles present. We'll take the second option here and let this run. And this will take just a moment, but when it's done, we'll get a table that again has 2,504 rows, 1 row per sample. But instead of 19,000 columns like we have now, we'll just have one column for each gene represented in the data. Within those columns, we'll have numbers representing how many variant sites were identified in each sample within the gene based on this filtering process that we used. So we'll give it just a moment. So here's our output. And with this sort of information now, we see most of the data is simply zeros. We have rare variance. We don't expect a lot of big numbers in here. But we do occasionally see some counts where we recognize, okay, a few samples have some variance in these genes. If we want to build out a simple burden test from this, we simply need to combine it with the phenotype data and apply some statistics. So SVS makes it really easy to do data management tasks like joining and merging spreadsheets. Here, with just a couple clicks, I can merge that variant counts per gene table that we created together with the phenotypic data. And with everything in one table now, I can select a dependent variable that I want to work with. In this case, I'll select the case control variable. And then I can select any of a number of different statistics to do an association test here. In this case, I'll just run the numeric associations, keep the standard default correlation trend test, let this run. It takes just a moment. And what I have now are gene by gene p-values, which, if sorted, I can see which genes come to the top. It looks like all of these first 4 were part of the simulation that we looked at a few minutes ago in the slides. But I can see exactly what the significance level was there. And if I want to go a step beyond and look at it visually, SVS makes it easy simply to click, put the p-values into the browser where I can see the gene by gene p-values. And beyond that, I can add to the visualization the exact data used in the analysis. So we have this final list of filtered variants that came through our filtering process. I can create a variant map from that. Then we can sort based on the case control status. And if we look at TPS-G1, which was our strongest result, what you should see here now is that within this image, most of the variation is in the lower part of the plot. The samples have been sorted so that cases are at the bottom, so the case equals true variable is represented by green on the axis. So we can see that we have a lot more variation building up in cases than controls. This is exactly what we're looking for with this type of a burden test. And if we look around at some of the other results, we should see similar patterns. So nearby at TPS-AB1, where we have a more moderate association, we see again most of the variation is in the lower part of the image. So with that, we've now set up a very simple, very basic burden test just by cobbling together a handful of features, a few data filters, some summaries, and finally a simple statistic. But what if you want to use one of the more formal association tests here? If we come back to the project, I went through and did this in advance for the whole exome. And we have results here ultimately showing everything right down through to running SCAT, KBAC, and a variant level association test. And just so you can see what the final step in the process looks like, when you have the case control variable together with all of the input variants you want to use, in this case there were about 400,000 variants across the exome that were included in the test. Then from the DNA-seq menu, under collapsing methods, you can choose if you want to run SCAT-O, if you want to run KBAC with one of the different testing options or CMC test. If you select SCAT-O as an example, you'll be prompted to choose the marker weighting scheme that you want to use. Choose the test if you want to run SCAT-O or a generalized SCAT test or both. The generalized SCAT test with the row parameter equals 0 should give you just the traditional basic SCAT test. moving down from there, you can make some choices about variants to select, regions to collapse in, if there are any covariates that you want to adjust for in the model, and proceed then to run the test. We won't do it right now. It takes a bit of time on this scale of data. But if you look at the results, the SCAT results, for example, will show you a gene name together with a p-value per gene. If you want to take a closer look at those, of course, you can sort, see what comes to the top. Recognize again here, it looks like our first 4 results are all from the simulation, and moving down we see some of the other simulated genes, TPSD1 among them. And if we look at it visually, in this plot now we'll see, go ahead and zoom out to the whole genome level, all of those p-values. So in green, what it's drawing in right now is the individual SNP level associations. In a moment, it will update with the KBAC and SCAT tests, and we'll start to fill in the whole exome variant summaries. Let's just take one of these genes and take a closer look at it here. So here as we look at COL26A1, it's a great example where you can recognize that the individual variant associations are not very strong. The best one here we see has a p-value of about 3 times 10 to the minus 4th, which is not bad, but it's not something that you would immediately recognize if you were doing a GWAS type of a test. But across the gene, all of these weakly associated signals turn into a much stronger signal at the gene level. So here we have a SCAT result with a p-value on the order of nearly 8 times 10 to the minus 9th. And KBAC p-value is actually maxed out at 10 to the minus 6th. We only did 1 million simulations on this with the permutation testing procedure, so that's the most significant possible result for KBAC. And as we look at some of the other results around the exome, we'll see a similar pattern. If we move back over to chromosome 16, where we were looking earlier, at TPSG1, here we see essentially the same pattern we saw with our simple burden test that we put together a moment ago, where we can see the results of the collapsing tests together with the results of the SNP by SNP tests and recognize the variation pattern that drives it. So with that, I think we can go ahead and close out of SVS for a little bit and go back to the slides and talk about what comes next.
So, what we were just looking at was ideal conditions. We knew exactly what a causal variant should look like and only included causal variants in the test. And in that situation, what we find is most of these came up pretty well. There were about 18,000 genes that were tested. If you assume a Bonferroni correction there, about 3 times 10 to the minus 6th is where you would declare significance. So with KBAC, 4 of the 8 would have been declared significant. With SCAT, only 2 of them, but definitely there's a trend in the right direction. The interesting thing is to look at the difference between SCAT and SCAT here, where SCAT just did not perform as well. You'll remember in the earlier slides we discussed that the SCAT test does not perform as well when variant effects are unidirectional. It turns out with our data simulation here, everything was simulated to be a unidirectional effect, and I believe that's what was holding SCAT back. Now, if you look at the tests directly against each other, so here we have each dot representing 1 gene, and the log 10 transform of the SCAT p-value against the SCAT-O, or the other way around. What you'll notice is in red are the 8 simulated genes. All 8 of them are among the best results in SCATO. It did a pretty good job of identifying the strongest signals, even if some of them failed to reach our significance threshold. Compared with SCAT, you'll see at least 5 of these are just buried down in the noise where you probably wouldn't have picked them out of a list. Again, I think that has a lot to do with the way the simulation was run, but it also is a reminder that no one test is necessarily going to be best in all situations. If you compare KBAC against SCATO in the same way, first thing to keep in mind is KBAC is based on a permutation process. The way it was run here, 10 to the minus 6th is the most significant possible p-value, so we have 4 of them that are maxed out. But generally speaking, they both performed fairly similarly in identifying our genes of interest. Even the genes that had less than 2% of all of the cases carrying a variant. In this case, where there wasn't too much noise in the variant data, it's still able to put those at the top of the list even if they're not reaching statistical significance. Now, what happens if you add some noise back into the data? So our loss of function model was just rare variants that were predicted damaging. What if we just bring in all of the rare non-synonymous variants, even the ones that are probably benign? Now, with some of the genes, it didn't make any difference, like COL26A1, all of the same variants were still there. But with others, it made a bigger difference, like IL4R. It doubled the amount of variation there. So there's 39 variant sites in the gene. Only 20 of them really contribute to the disease. The significance was cut in half. If you compare these p-values to what were in the previous slide, it really makes a big difference. And some of the methods are going to be better at cutting through the noise than others. Different way here. This time, instead of just putting in more rare variant noise, this was adding in more common variant noise. And again, with some genes, it didn't make much difference, but one of the glaring examples to me is TPSG1. This gene, SCATO, when it just had the basic simulated loss of function variance, it was 10 to the minus 10th. Here it drops down to much, much lower significance. And I think a lot of this has to do with the way common variants are handled. Notice we didn't use KBAC here. We used CMC, which is the one that is able to handle common variants a little bit better. It also didn't do well with this gene. I think there were just too many variants going in the opposite direction of the effect to handle it. But you'll also see some of the other genes still were detected relatively well. Now, just a more direct comparison in this case of CMC versus SCATO, I want to point a few things out. Number one was this result here at the upper left. This was the 4th and 5th best genes in CMC. They happen to be overlapping genes. One of them is, let me check my notes here, ST20. What happened here is that the signal was driven primarily by a very common variant in ST20. And I believe that the reason why SCATO pushed it so far down is because of the weighting procedure used in SCATO. If I had used different weights there, then that gene may have been more significant in both methods. On the other hand, you look at something like this variant over here to the right, this, or not variant, but gene, it's RSPH10B, which was the 2nd best overall result in SCATO, but in CMC, it's down quite low in significance. The interesting thing here, when you go look at it, is that there were several influential variants in both directions. There's one especially influential variant that was predominantly in controls rather than cases. The SCAT family of methods are pretty good at finding those genes where there are variants that work in both directions. Some of these burden tests like CMC are not quite so good at that, and that's why we see results like this. So again, there are different factors to consider, different hypotheses you might want to test as you're working with rare variant data. So let's go ahead and move on. To this point, everything that we've been talking about is firmly rooted in what we typically call the tertiary analysis phase of sequence analysis, the sense-making portion. Quite often when we're working with customers who are doing this, they have a lot of questions about secondary analysis. What do you need to know in terms of variant calling? What do you need to know in terms of quality assurance procedures when you're doing this kind of work? And I have just a couple of slides here. First of all, what did we do in GWAS? Remember, GWAS is all about common variants, but we would look at things like call rates at a sample and SNP level to determine quality. We'd look at things like Hardy-Weinberg equilibrium and minor allele frequencies to determine which SNPs are suitable for testing. Of course, those things don't work as well for rare variant analysis. We have to look at different types of quality statistics. And in NGS, you see a lot of different things being used. You can look at coverage depth either at individual variants or averaged across entire samples to determine quality. You can look at different sorts of quality scores per variant or per individual genotype call. Those are very helpful. There are sample level statistics like singleton counts or transition transversion ratios that help us to identify outlier samples. Region mappability is useful for identifying problematic variants. But with all of this, what I really encourage you to think about is consistency. This slide is a couple years old, but I think it still holds up pretty well. What happens with rare variants is that when you really think about the variant calling process, the rarest variants are also the most likely to be incorrectly called. If a variant is called that is something that is very common in your population, it's a pretty good chance it's really there. If it's something that's never been seen anywhere before, then it's something that you might need to be a little more concerned about. And this image is based on some work Golson-Lyon did a few years ago, running the same sample through 5 different variant callers. When you look at variants that were known in a particular version of dbSNP, the concordance between the 5 callers is pretty good. When you look at the variants that were completely novel according to that version of dbSNP, the concordance goes way down. And that's because each different variant caller has its own rules for how it handles particular edge cases. Some things are going to get called in one and not in another, and so on. And the implication for rare variant analysis is be consistent, especially when it comes to selecting control groups. You might be tempted to use public controls, maybe 1,000 genomes, maybe ESP or otherwise. And I would suggest that when possible, it's best if you can find controls that were processed using the same bioinformatics workflows as your study samples, and try to keep things as consistent as possible, especially when it comes to rare variant analysis. So what about common variants? The question comes up fairly often here at Golden Helix from clients and potential clients. I've got sequence data. Can I use all of the GWAS functionality in SVS on these more common variants? And the question, or the answer, is yes. You can use a lot of the GWAS style functionality, but there are some caveats there. It's always helpful, of course, to have homozygous reference genotype calls in the data. We don't always get that with sequencing data. 1,000 genomes is great because they do have that. They have all of the multi-sample VCF files, so we know what those genotypes are supposed to be, because most of the functions you look at need to know heterozygous, rare, homozygous, common, and so on. And there are certain procedures that are going to require careful consideration in terms of variant selection. Things like principal components analysis or IBD analysis use methods that assume you have markers that are highly informative, spread evenly across the genome, and especially if you're working with exome sequencing data or data from other targeted sequencing experiments, that's not always going to be the case. But there are processes you can follow to at least simulate or approximate that sort of a situation. With something like IBD analysis, that's what the image on the right comes from. I was working with these exomes from 1,000 Genomes Phase 3, and you can identify that there are a handful of apparently first-degree relative pairs in that data, which is what we're seeing here. You just have to be smart about how you go about choosing the variants you use for that analysis. So one of the nice things with Phase 3,000 genomes is they have SNP data to go along with the sequence data. And I downloaded the SNP data and just did some comparisons and went through a marker selection process. On the left is what you might use with GWAS data if you were getting ready to do principal components analysis. So starting from the AFI6 chip, you've got about 867,000 autosomal SNPs. 806 of those pass some basic QC filters on allele frequency and call rate, do an LD pruning process, and you get 74,000 fairly evenly spaced common variants. With the 1,000 genomes Phase 3 data, you've got 81 million variants in the autosomal data alone. I was focused just on exons plus or minus 5 base pairs. I had filtered the data in advance. So there's about 2.2 million of those. But if we follow a similar process and just look at the common ones, so greater than 1% frequency, and do LD pruning, we get a similar number, although they're very different. Out of these 74,000, only 1,000 are in exon regions. So there's a chance these are going to pick up different things. But if you take those two sets and run them both through principal components analysis, the result is stunningly similar, at least in the first two components. What you should see here is essentially a reflection where the first two principal components based on the SNP data or based on exon data, it's pretty much identical. They're both picking up the same high-level population structure in the data with the African populations, the European, the East Asian, South Asian, and admixed Americans. It's just the first component is reversed, which really doesn't make all that much difference. They're detecting the same patterns. Now, the question then becomes, though, do they really detect the same effects in the data? And here, if you look at the first component based on the AFI data or based on the exome, it's a very strong linear relationship. The second component, again, very strong linear relationship. They're picking up pretty much the same things. But if you move beyond that, the third component, they're starting to diverge from that linear relationship. Fourth component, diverging a little bit more. By the time you get to the fifth component, they're measuring completely different things. So it's important here to keep in mind that the variants within coding regions are under different selective pressures than the variants in the genome at large. And so if you want to use some of these methods, you need to carefully consider what it is that you're really calculating and determining. But if you just need to adjust for population structure based on the first 2 or 3 components, you can usually do that with exome data. So, in conclusion, what have we learned here today? Number one, SVS is a comprehensive platform for analysis of both common and rare sequence variants. There's a lot of power for the rare variant association tests, SCATO being the newest of those. SCATO, as we saw, is an optimized combination of SCAT and burden testing approaches. The power of those tests, though, is affected by many different factors. the variant weighting scheme that's used can really influence the results. The upstream variant filtering and selection process is very important. In reality, we don't know which variants are going to be the causal variants. We just have to kind of guess what that profile looks like of a causal variant and hope that we're picking up the signal. Sample size then becomes very important. So the larger your sample is, the better chance you have of getting past that suboptimal variant selection in terms of finding the right spots. And of course, the true underlying biology of the disease is always the real question here that we're trying to solve. So with that, I hope that you've enjoyed the presentation today. I hope you've learned something. If you have any questions or more, would like more information, I think Cheryl has some information for you. Cheryl, are you still there?
I am, Bryce. Thank you. I will have the recording and the slides up by the end of the week. And we are going to take some questions. If you haven't answered those, feel free to put them into the questions pane. I do see quite a few questions, and we only have a few minutes left, so trust that if Bryce doesn't get to them, we will get in touch with you to make sure that they are addressed. With that, Bryce, I'm going to turn it right back over to you so that we can get those questions answered.
Okay. So I will try to answer as many of these questions here as I can. We've got some really interesting questions that have come in along the way. First question, how do I create an input file for SVS? The primary supported input format here is going to be a VCF file. All of the examples that I showed today were based on reading in VCF files directly from the 1000 Genomes project. I expect that most of the time if you have sequence data, you'll already have that format. Now, if you don't have VCF files, there are a number of other supported formats. Of course, we read in a lot of the standard things from GWAS programs like PED files or the Plink binary version of PED files, or just standard flat text files. So a lot of options there. Another question, can I use SVS to call variants? The answer there is no. We don't do any variant calling. We expect that you have done that upstream and you're coming to us with called variants. Next question, how CMC combines rare and common variants and prioritize variants for follow-up. So the way CMC works is that it initially requires you to set some thresholds for the minor allele frequency to create frequency bins for each variant. So the method that I used in the examples was to set 5 different frequency bins, less than 1%, between 1 and 2.5, 2.5 to 5, and on up. There are some different groupings. But it does a collapsing test within each of those bins and then has a multivariate statistic that it uses to determine the total significance across all of them. Now, your question, the second part of it, is prioritizing variants for follow-up. CMC only gives the gene level p-value. So if you need to prioritize variants within the gene for following up, what I would typically do is go back and do a variant level association test for the variants within the gene, determine which, if any of them, have the strongest association. Typically, though, you would expect that several of them should be followed up on, and you can always look up the annotations for all of the variants, determine the population frequency for all of them and other functional bits of data. And there's a similar question related to SCAT-O. Since the results are at a gene level, how do you prioritize the SNPs within genes? Again, typically you won't have more than maybe a few dozen. It depends on your sample size. You might have 5 variant sites in the gene, you might have a few dozen, but none of them typically are going to be carried by very many individuals. So it may be that you need to follow up on all of them. I would definitely, at a minimum, look at all of the variants contributing to the test go back and look at their functional data, look at their frequency, and determine which ones might have the greatest impact on the protein structure. But again, you should probably be looking at all of them. So moving down, how much is the overlap when different rare variant analysis methods are used? And that question came in fairly early in the process. Hopefully you saw that in the comparative plots that we did. The methods in some cases are very similar to each other. Sometimes they're quite different. And as explained, that has to do with the fact that they're not all testing the same hypothesis. The SCAT family of methods are better when you have bidirectional effects. The burden methods are better when you have unidirectional effects. In this particular simulation, where everything was simulated unidirectional, we had some very similar results, but that won't always be the case. Does 1,000 genomes phase 3 data for all chromosomes available in SVS now? That's coming. We're working on updating a lot of our annotation sources, and keep your keep watching. It'll be coming out soon. Can we use this analysis procedure to analyze GWAS data? That's another question that comes up from time to time. So the reverse of the question proposed in the webcast of can you use GWAS methods for the sequence data? Can you use sequence methods with GWAS data? And I hesitate to say too much there. Certainly within the software, there's nothing that will prevent you from doing so. And a few of the methods that allow for incorporating more common variants will probably give you some useful information in terms of a gene level test statistic, but it's not something that I have a lot of experience with and haven't spent much time really looking into what you'll get from that. Another question, does SVS contain whole genome sequence pipelines so I can include FASTQ files and get out VCF? Unfortunately, no, we don't do alignment or variant calling right now. Another question, LD pruning for rare variants. Are rare variants in LD? So that question would have come in about the time that I mentioned LD pruning. And keep in mind that the filter that we used there was to first reduce only to variants with an observed frequency greater than 1%, which with the sample size we have indicates a very large number of individuals carrying it. We should be able to detect LD at that level pretty well. Now, of course, at lower frequencies, those variants are rarely going to occur together with anything else, so it's going to be difficult to calculate an accurate linkage disequilibrium statistic. How do KBAC, SCATO, and CMC work when analyzing common variant association? Which one is best? I will say in that regard that CMC is the one that's really designed to handle common variants the best. KBAC is strictly a rare variant test. SCAT, I haven't had enough experience with to really say how it's going to behave with common variants, but the default weighting scheme will really penalize the more common variation unless you use an alternate weighting scheme. This is an interesting question. What is the variance explained by the fifth component in the exome versus AFI data? Let's go back and look at that because it is an interesting picture. What you see in the AFI data, the blue groups are the African populations. That fifth component primarily is separating the different subpopulations within the African superpopulation. So if you were just to look at that univariate, perhaps as a histogram, you would see that that fifth component is 5 major clusters. While if you looked at the exome data, univariate as a histogram, it would be more of a smooth curve that's detecting some other difference that is more uniform across all of the samples. They're clearly detecting different effects. I might have to dig into it a little deeper to learn exactly what it is. Now, continuing the question, if the variance explained is very low, would you be concerned about the very different outcomes for the comparison? That's an interesting question. In this case, I wasn't interested in using the components for anything except just to define the population structure. And it was, that's a question we can take offline here. Okay. How do collapsing tests integrate study designs that involve related family members, such as trios or greater with affected and unaffected family members? That is a really interesting question. And the tests that I showed here today are all tests that assume population-based study designs. They're not going to account directly for family-based data collection. That being said, if you look around in the literature, there are alternate versions of some of these tests. Like I think there's one called SCAT-FAM that's designed to incorporate some sort of a transmission statistic within families. Within SVS, your best option would be to use the KBAC, so the mixed model KBAC, where you can incorporate a kinship matrix to the analysis. What it will do then is correctly adjust the test, recognizing that some samples with the same variant happen to be related to each other, although it won't necessarily incorporate any kind of transmission or linkage information there. Okay, so last question, how useful and representative of extreme phenotypes for rare variant discovery to the general population? That's another interesting question, and most of the methods that we've talked about here today are designed for binary phenotype outcomes. If you are looking at quantitative traits and extreme values, that is an acceptable use case, especially if you convert it into more of a dichotomous outcome, comparing the really extreme cases against the general population. That should work out pretty well. So with that, I think we'll go ahead and wrap up. I'd like to thank everyone again for coming today. And Cheryl, do you have any final comments before we sign off?
Bryce, I just want to thank you for your time. Questions that were not answered will be answered, and we'll be in touch with you. If you have any further questions outside of the ones that were asked, feel free to reach us at any point. Thanks so much, everyone. Have a great day.